using stem cells for human therapy, cell replacement, drug screening, and disease modeling. I'll give you a little brief bio. I'm just looking through there. Dr. Sullivan was born in Cork City. He graduated from University College Cork with a degree in biochemistry. Following this, he went on to study for a master's in research from Trinity College Dublin. He then went on to the Rosalind Institute in Edinburgh, where he earned his doctorate under Professors Jim McQueer and Ian Wilmot, which are two of the scientists responsible for cloning dive sheep. There, Dr. Sullivan became one of the first researchers in Europe to culture human pluripotent stem cells, and you'll find out more about this for Dr. Sullivan. He moved to the University of Cambridge, where he got an Isaac Newton Trust Fellowship. He subsequently went on to work as a research fellow at the Harvard University, where he earned a stem cell research fund scholarship. So there you go, that's his bio, and I'll hand you over to him now. So.
First of all, just let me say thanks very much for coming in when the weather is so good outside after the last 2 Irish summers. I think you're making a specific, a very, very big sacrifice to come in when the weather's this good. So thanks very much. I've been back in Dublin for the last 6 months now, and it's really very encouraging. I've been away for a long period of time, and coming back to Ireland now, it's really striking how much more people are becoming engaged in scientific topics in Ireland now. You've got, like, for example, this ALCHEMIST Cafe. You've got things like Science Spin magazine, and also you've got a lot more science on the TV. And what I really like is the Science Gallery, where Cormac gave us a great talk on the Big Bang last week, or 2 weeks ago. But the Science Gallery is specifically designed to bring science to a broader audience, because I think in this day and age, scientists, it's not always clear to people why scientists want to do certain things that they want to do. And this is particularly in my field of stem cell research, where there is, every day there is like a different story or a different spin and a different misinformation kind of in the newspapers or in the media. And I wanted to touch on that towards the end of the talk, that really, media is changing from print to digital, and that big upset now is changing dramatically how we get our information. And when we're looking for professionals, it's not always easy to find the professionals versus some person who just likes to rant in an unsubstantiated fashion on the internet. And this is, I think, pertinent very much now, because we have a rising degree of stem cell fraud. You have people making money off people who are in desperate situations, who have either sickness themselves, or they have a relative who has a sickness, and they log on to a website, and the website tells them that there's a cure available, when I think the medical and scientific consensus is there is no such cure as yet. So excitement that really science is getting deeper into the public consciousness, and that's great to see in Ireland. It really is. So well done on the ALCHEMIST Cafe for taking part in that. My curiosity as a scientist is more or less centered on the regenerative medicine and developmental biology field. So I'm kind of interested in how a fertilized egg becomes a full sentient thinking human being, how cells know what to do at the right time. How does a neuron know to become a neuron? How does a liver cell know to become a liver cell? And the great orchestration of development is intriguing to me, that you can get such complexity arising from initially just one fertilized egg. So, also of interest is the idea of regeneration, how we regenerate ourselves. For example, like in our gut, for example, the gut regenerates itself almost on a daily basis. The lining that our food passes through and gets absorbed. And that's very, very interesting that, you know, when you take, say, for example, lower vertebrates like a salamander, if you cut a limb off a salamander, the whole limb regenerates. And, you know, there's this great interest in if the lower vertebrates have this capacity, why doesn't it still exist in higher vertebrates? And whether actually that capacity is still there, but something is inhibiting it. Like, for example, something like scar tissue formation interferes with some regenerative capacity that's already underlying. So these are really kind of big questions relating to either directly or obliquely to stem cells. Stem cells are a unique kind of cell that have two characteristics that make them extremely interesting to biology, both as a tool, but also as a cellular identity. They have the capacity not only to self-renew, so like one stem cell can become two stem cells, can become four stem cells, and so on. And this, I suppose, capacity to start off with a small amount of material and augment that material is very, very important for a lot of kind of experiments that basic science wants to do, but also is extremely important for having enough material to effectively treat a patient with a disease. So, self-renewal. The second capacity of the stem cells that make them remarkable is that they can also form cells fated to different functions. So, for example, if you take a blood stem cell, known in the trade as a hematopoietic stem cell, it can divide to give you more hematopoietic stem cells. So you start off with a small number of cells, and you can augment that number. But also, they can make all the cells of the blood system. So, you know, this is extremely. These two characteristics make them extremely useful for, say, conventional stem cell therapies involving leukemia or rare blood diseases. Like, for example, way back in the atomic bombs landed on Hiroshima, there was a lot of interest in that. You know, apart from the initial blast that killed lots of people, subsequently people died a few weeks later. And what they basically came to understand is the radiation was killing fast proliferating cells that were essential for the survival of the people. So they did more research into this and did several very interesting experiments with mice, where they took bone marrow from a normal mouse and then transplanted that into an irradiated mouse, and they found that the irradiated mouse was rescued. It didn't, after being exposed to the irradiation, die anymore. And we now know that is because there are several stem cells present in the bone marrow that replaced the cells that were initially damaged and died after the mouse had been exposed to radiation. So these stem cells are hematopoietic stem cells or blood stem cells, and also mesenchymal stem cells. So between the two of them, they repopulate a system like the immune system and the blood system, which are essential for continued survival of the mouse. So, in many ways, stem cell therapy is not new. It's now 50 years we've been transferring bone marrow cells into patients in one form or another, and even now, after 50 years, the number of diseases that we can cure and treat is still very small. And the reason for this is that there are a lot of questions that are still unanswered in stem cell biology. One of the things is that stem cells, such as adult stem cells like blood stem cells and mesenchymal stem cells, they're very hard to find. They're very hard to isolate from other cells that look sort of similar in the bone marrow but don't exhibit the same properties. And this is very much the quest in adult stem cell biology, is to develop better systems to identify the stem cell that has the regenerative capacity and be able to grow it in culture to expand the amount of material, and then to learn how to be able to coax that stem cell to become different cell types that we might require to cure a patient with a condition. So adult stem cells are resident in the adult, but they have limitations in that they're hard to identify. And if you are successful to identify them, even to extend their purity. To reiterate, stem cells have the capacity to self-renew, so you can increase them in number, and they have the capacity to form different cell types. As you alluded to there, the limitations of most forms of adult stem cells is they're difficult to find, and furthermore, they're not easy to propagate in culture. So if you get a population of cells where you say where there are some stem cells, you mightn't be able to expand them in culture to a number that would allow you to do a well-controlled experiment or to be able to treat someone effectively. So each different, like there are 3 types of stem cells. For that I went 3 classifications I'm using for the purposes of this talk: adult stem cells, embryonic stem cells, and what are now called induced pluripotent stem cells. Adult stem cells are the only stem cells so far that have been used successfully in a direct clinical context. Embryonic stem cells are cells that are derived from embryos, and the embryos would be surplus to the IVF process, and they would be received from couples who decided they'd had enough children, or they just decided they wanted to donate those embryos for research and medical purposes. They would also have other options like putting them in the freezer or throwing them out as medical waste. The way the medicine works, when a woman goes for IVF treatment, she's given drugs to make her super ovulate or release eggs into the fallopian tube, and these are the drugs actually don't work, or they work very well. So you frequently get around 8 or 9 eggs, and in order to maximize the chance of the woman getting a pregnancy, what is done is that all the eggs are taken and fertilized in a dish with sperm. So they're fertilized outside the woman in a petri dish, and then usually 2 or 3 would be reimplanted back into the woman, and hopefully they would implant, or 1 or 2 of them would implant into the uterus of the woman, and then subsequently further development occurs. It is important to realize that if those embryos do not have access to a womb, they divide a few times and then expire, because it's a symbiotic relationship. Like in order for a pregnancy to persist, an embryo needs to be embedded in the womb so that it can receive the signals that it needs to continue development and then form a more like a ball of cells that differentiates to the extent that it starts having brain cells and other different types of other types of cells. So, in the context of embryonic stem cells, we're talking about a sperm and an egg that have been fused in a dish and have let divides over 5 days in what's called a blastocyst. And what you do when you're deriving embryonic stem cells is that you remove a portion of these cells and then put them in some food, soup for want of a better word. And some of those cells acclimatize to their new foreign environment and start to grow and proliferate. And then those cells, you can continue proliferating for ages. And then you have what's called an embryonic stem cell line that can be frozen in liquid nitrogen and resuscitated, and then it can be differentiated into all the cell types of the human body. Now, the crux, why people are so excited about these particular type of stem cells is there are many diseases for which there is no stem cell available. So for example, diabetes, there is no adult stem cell in the pancreas. So, or you take diseases like neurodegenerative diseases, we still have not been very successful at being able to isolate neural stem cells or culture them outside in the petri dish. Embryonic stem cells, on the other hand, we can very easily generate different types of neurons from them What this represents is that it's alleviating a scarcity of material for basic research purposes. I'll give you an example. Say, for example, you have a patient with Parkinson's disease, and I'm the clinician trying to treat the patient and trying to basically develop a better treatment or even reverse the disease, Parkinson's disease. If I go to the patient, at the moment I don't know whether the change that caused the emergence of his or her Parkinsonian symptoms happened last week or happened when he was a teenager years ago. So really, at the moment we're arriving at a crime scene, and we have no idea when the crime took place, and we have no idea whether, you know, the knife on the table was involved in the crime. Maybe it was placed there, the crime was like several years ago, and the knife only appeared last week. The interest in stem cells is such that we can take a stem cell that is fated to display Parkinson's genetically, and then we can put it in a different soup to make it stop dividing as stem cells, but rather differentiate to become a particular type of neuron that's lost in Parkinson's, so in this case, dopaminergic neurons. So scientists are interested, and clinicians are interested, in embryonic stem cells, not because we love controversy, it's because they have characteristics that other stem cells do not have. And when you take diseases like diabetes, neurodegenerative diseases like Parkinson's, Alzheimer's, there's a huge range of diseases that traditional methods for trying to understand how those diseases occur have, despite enormous effort, have failed us. We know relatively little, for example, about the triggers of Parkinson's. So really, the argument is about like embryos that will never be implanted back into a woman, will therefore never go forward in development to become what's called a post-implantation embryo. So they will never become a pregnancy. And in Ireland at the moment, we have a legal vacuum where there are huge numbers of embryos sitting in freezers, and they will never be used for any purpose. At the moment, couples cannot donate those embryos to other couples. All you can do is use them to treat your own infertility and try and get pregnant. But there is all these extra excess embryos that are basically sitting in freezers in Irish IVF clinics that will eventually be thrown out as medical waste because there is no legal framework with which to do research or donate those embryos to other couples who have infertility problems. So you also have heard of the third type of stem cells. They've been all over the RTE and the Irish Times and all over the world's media. And these stem cells are called induced pluripotent stem cells. And what these cells are skin cells that have been heavily manipulated so that 4 key factors are introduced into those cells, and their expression is forced at a very high level over a long period of time, typically, 6 weeks. And what happens when you force the expression of these key factors, it alters the cell in such a way that the initial skin cell or the initial somatic cell that you had acquires characteristics that up to now would only have been seen in pluripotent stem cells. And there's huge excitement about this phenomenon because number one, it might be that we can reprogram cells to an extent where I could take, for example, a patient's skin cells and reprogram them back and then coax those cells to become the cell type that the patient needs in order to alleviate the pathology of the disease from which he's suffering. The world media grabbed on this, and I think they transmogrified the honest enthusiasm and surprise that the biological and clinical communities had that this could ever occur, that you could artificially, just with 4 factors, restore such characteristics to a cell. They transmogrified that surprise into stories that said embryonic stem cells are no longer needed. And anybody who's a serious scientist will tell you there are striking differences between human embryonic stem cells and induced pluripotent stem cells. Even looking down the microscope, they look slightly different. And when you subject them to functional tests with which human embryonic stem cells are differentiated from other aberrant cell types, the induced pluripotent stem cells as yet don't pass all those functional tests. So really the take-home message with induced pluripotent stem cells is that this is an unexpected result, that we could make a cell forget what it is in such a simple manner. However, whether it will be clinically relevant is still very much up in the air. And anything that you read and anything that you see on television telling you that you don't have to make a moral decision about embryonic stem cells because induced pluripotent stem cells are now there is a falsehood. The evidence that is there to date does not substantiate the supposition that human embryonic stem cells are no longer needed for understanding basic aspects of human development and treating people. Embryonic stem cells are de facto years closer to the clinic. So you have, for example, now that good old George Bush has finally left the building, a roadblock has been removed from clinical trials. And clinical trials are now starting with derivatives of embryonic stem cells. So for example, there are 2 clinical trials that are making the media at the moment. One is differentiating the cells into something that will become a neuron precursor, and that's being used, and factors from that cell are being used to try and alleviate aspects of spinal injury, and that's being done over in California. And then there's another disease, there's an age-related macular degeneration, which is a blindness disease, which Senator David Norris has, and there's a clinical trial there that's just started, that's just in the process of gearing up to start, where they make the cells that are lost in the disease from embryonic stem cells and then implant them on a membrane and then put them at the back of the eye where there has been degeneration of a specific cell type. And the idea is to put healthy cells back in, remove the cells that are no longer functional, and that would alleviate the blindness suffered by patients of this disease. So I think it's very important to stress that any serious scientist or clinician, if they want to reduce the amount of time between their research and somebody having a clinical benefit, they will use all the different cell types at their disposal. Each different cell type has different characteristics. Depending on what biological question you're trying to address, you'll use a different cell type. Pluripotent stem cells, i.e. embryonic stem cells and induced pluripotent stem cells, are enjoying a lot of media attention at the moment, but they're being misreported. So we have stem cell biology, which is basically, it's developing week on week. If there's not a very significant but small step forward every week, there's some political controversy, or there's some tale of a person who's been given misinformation going off and making what would be considered by any specialists in the field a completely inappropriate medical decision. So really we're at the stage now where we have an emerging technology that is coming out and generating huge amounts of interest. And we've also got a profound change in the way that people gather their information. So you also have, I suppose, people going abroad for treatments and not differentiating between what would be considered a reputable I suppose, medical centre and a medical centre that doesn't seem to have good credibility. You know, like these clinics in China, for example, who are offering patient testimony as evidence that their particular stem cell therapy works. It is important to remember that the basic psychology of someone who has a disease and is desperately wanting to cure themselves, they're desperate. They will try anything, and they will believe in anything. So really what we have to do is give them critical and balanced medical information by which they can, I suppose, have a better idea of the risk inherent in the treatment that they're going for. And that involves wider education, and that involves bringing these kind of subjects out into the light and shining a light on them and debating them more, I suppose, more forcefully. So I think we'll stop there and maybe get some more questions.
Yeah, there's actually food's actually being served at the back.
Oh, okay.